All right. Welcome back, everyone, to this Zebrafish Global Educational Network. My name is Jean-Pierre Peron, and I'm Associate Professor of Immunology here at the Department of Immunology, University of São Paulo, Brazil. And I have to say, it is a huge pleasure for me to share this session today with you. So the talk today is going to be performed by Dr. Loïc Royer. So Dr. Loïc, he had his PhD in bioinformatics at Dresden University, and then he was involved in the development of a beautiful platform of self-driving multi-viewing light sheet microscope while he was at the Max Planck Institute. Dr. Loïc is now part of the Chan Zuckerberg Biohub, and he is fascinated in understanding how organisms develop from single cells into fully functional self-organizing uh, tissues. To solve that, he is dedicated to develop novel and pioneering strategies for imaging and imaging processing. Such, of course, the one that he's going to show to us today, the beautiful work on multimodal atlas of vertebrate development with light sheet and single embryo single cell RNA sequencing. Dr. Loïc, it's a huge pleasure to have you here with us. Thank you so much. The stage is yours.
Thank you so much for the very kind introduction, and thanks to all the organizer for inviting me today. I'm very happy to be able to present my work today on this the work of my team and of the Biohub here today. As was explained, I'm going to talk about one of our latest projects that combines light sheet imaging with single cell sequencing, and really the thing we're trying to do is trying to understand how an organism is built. And so, as a quick introduction about the Biohub, we are a non-profit research institute located in San Francisco. We have a kind of we are a hub between UCSF, Berkeley, and Stanford. And within the Biohub, there's an intramural research program of which my team is part of. My team is really made of three of individuals that span three disciplines. I have engineers, mostly optical engineers, computationalists that are, for example, software engineers, computer vision experts, people that can, process images and turn images into knowledge. And of course, my team also has biologists because we're trying to use these technologies that we're developing to actually answer fundamental questions. And the area of biology we're interested in is developmental biology, and the model organism that we chose is Zebrafish. So there's this quote from Sidney Brenner that I really like, which is, "Progress in science depends on new techniques, new discoveries, and new ideas, probably in that order." And really what's really powerful about that quote from Sidney is that it really shows that what the starting point must be new technologies, because new technologies reveal what's not visible, what was not visible previously, and this really helps opening up the opportunities towards new discoveries and new ideas. And so, as I started my lab 6 years ago now, we started to develop a lot of technologies to help us in our quest. So the first thing we did was to build a fish facility, and for that we actually rethought the way that fish are reared, and we built a fully automated and compact fish facility. But we also have designed and built advanced light sheet microscopes, and I will show some of these results today. And that's very important because, again, we need to be able to see better the processes of interest. And with some colleagues here in San Francisco, but also in Australia, about 4 years ago, we started a project called Napari, which has been revolutionizing how images are visualized in Python. And I learned recently that we have about 10,000 downloads every month on this. And it's really, if you like Python, if you use Python as a scientific programming language, you want to check Napari as a way to visualize and analyze your images. We've been writing a lot of code, GPU accelerated, to be able to process the terabytes of data that our microscopes are generating, to be able to massage these images, to make them better, to make them, and to basically extract insights from it. And one of the key things that we need to do is to be able to track cells in space and time, and on the basis of our light sheet data. And so that's been that has been definitely an effort that we have put a lot of work on. We also have been designing tools like IDEN that allow biologists to very easily denoise images. And this is based on ideas from deep learning and self-supervised learning, and we really try to make a tool that's extremely easy for people to use, so they can denoise images in a very convenient manner. And then more recently, we also had some very nice work published where we use deep learning to featurize images from cells. That's a little bit more a tangent from the work that we'll present today. But yeah, today I will focus more on our work on imaging and also on sequencing. But all these technologies that I just show here really are kind of the foundation from which we could do all the work that I will show you today. And as I said, this is all a means to an end. All these are techniques that we had to develop as a means to an end, and the end is the biology, right? And the dream we really have is, and I think that's the dream that the whole field has, is really to try to understand organisms, developing organisms such that we can, follow the cells in space and time, so the dynamics of the cells, but also understand the lineages and the fate of these cells. And ultimately, and ideally, we would also want to be able to very well understand and map the identity of these cells and their state as the organism develops, as the tissues unfold. So towards that grand goal, we have to have first steps. And so I will present today a project where we focused on a very, very particular question, and we used all these technologies we developed and we refined them using that biological question. And that is what I'm going to talk today about. So, but first, let me go back a little bit to the basics and refresh people's minds on development and some of these on the key basics that are required to understand what I will discuss today. So remember that gastrulation is the conserved process leading to the formation of 3 germ layers. This is one of the very basic tenets of developmental biology. This is a very conserved process. Humans undergo gastrulation. Fish also undergo gastrulation, and many other species do. And these 3 germ layers that are basically arise after gastrulation are the ectoderm, the mesoderm, and the endoderm. The ectoderm is essentially the nervous system and the skin. Mesoderm is bones and muscles, and endoderm are internal organs. And so the theory really, the assumption and kind of the textbook, statements there are that past gastrulation, these 3 germ layers are segregated, and so you have basically committed lineages of multipotent progenitors, and they're not supposed to be cells that cross these boundaries. Okay, but is it really the case? And so there's been really, so that's kind of the textbook picture, but things are a little bit more complicated. And there's been quite a lot of evidence, starting in 2009 with some work from the team of Jean-François and Nicolas, that showed that actually maybe there are actually cells that are very late in development, post-gastrulation, actually still have descendants that go both in the ectoderm and the mesoderm. And since then, in different species, the same results and the same ideas have been coming up. And so there is clearly now evidence that there are such cells, but they are still not fully characterized, I would say. There's still a little bit of mystery surrounding them. So, and then even more recently, a lot of people have been working on these things. And in particular, we have been, mentored by Olivier Pourquier, who has also recently published something in eLife with his then postdoc Charlène Guyot. So So we found that question very interesting, trying to understand these NMPs, and we wanted to actually dig deeper into this topic and understand and help the field and help this community of people working on this to see if we can bring something to the table and help shed some light on this process. So it seems that there are such cells that are late pluripotent progenitors, right? And so what does that mean is that these cells would then essentially have the descendants that would go both to the neural tissue, but also mesoneurial tissue. So there will not be this segregation assumed. So the question is really where are these cells located in the embryo? So if you look at a 19-hour post-fertilization zebrafish embryo, really these cells are supposed to be towards the tail. So if we look a little bit at the anatomy, you have the somites here, you have the neural tube, the presomitic mesoderm, and the tail bud. And what we know is that there are progenitors that are neuroectodermal that will give rise to the neural tissue there, but we also have mesodermal progenitors that are in the presomitic mesoderm that will eventually that are in the tail bud and will give rise to tissue in the mesoderm, and particularly in the presomitic mesoderm. And really these NMPs are and we know they are located in the tail bud. And so what would happen is they would eventually divide, and some of these descendants would go into the neural tissue and others will go into the mesoderm tissue. So that's really like the setting, right? However, we've never really seen an NMP really undergo that trajectory all the way from the tail bud to dividing and then descendants integrating into these two tissues. And one of the reasons why it is difficult why we haven't seen that yet, perhaps, is that it's actually a very dynamic tissue. The tail bud is very dynamic. Cells are moving very fast, lots of divisions. And when we look at the whole and our data, and we look at the whole tail, this is a region that is the hardest to track, because that's where the cells are moving the fastest and in the most chaotic fashion. So instead of going straight into imaging, as we are kind of experts at, we actually decided to take a tangent and leverage the amazing capabilities at the Biohub in terms of single-cell sequencing. We have colleagues at the Biohub, Angela Pisco and Norman Neff, who are leading the data science team and the genomics team that are really experts at this. And so we teamed up with them and their teams, and we started looking at if we could do single-cell sequencing to understand better the process in the tail. And what happened is actually it went so well that we just decided to do more of it. The biologists in my team were very excited about doing single-cell sequencing, and then we actually decided to actually go further and see if we could do single-embryo, single-cell RNA sequencing. And so the result of that enthusiasm was really a large data set of single-embryo, single-cell RNA sequencing data over 10 stages, a total of 40 embryos, and a grand total of 120,000 cells. And we have put all this data in a website on zebrahub.org because we thought that it was useful for answering our own question about NMPs, or at least shed some light on it, and perhaps it would be useful for others. And so in the spirit of advanced science, which is something that's very important for the Biohub, we decided to release that even before our preprint would be out, so as to accelerate science. So let me dig a little bit into this as a parenthesis. The data set is really beautiful. It didn't. We could integrate all these time points together and really visualize the different stages of development. This is actually more visible in the previous slide. If you look at this video here, the different colors in the video reflect, and you see it right now, reflect the different stages of development, and you can really see some developmental trajectories arise in that UMAP. So that's actually, for those that are not familiar, this is a UMAP. This is a low-dimensional projection of this high-dimensional data set, which is where basically for every cell you have all almost the gene expression of every single gene. So it's a very high dimensional data set, and we use dimension reduction techniques such as UMAP to basically create this 3D representation of the whole data set. And we can also, so what we've done is we've been annotating this data set and basically identifying cell types within this data. And here you see some kind of grand structures. So these are clusters at a very high level, such as the mesoderm or some neural tissues. So here is a list of different tissues that we annotate. But we can go actually deeper, and we can get even more detailed and more kind of fine-grained cell types or groups of cell types that are related. So this very exciting data set that was very easy to kind of integrate into one data set. Remember, these are 10 stages of development. And for each stage of development, we have about four embryos, and it was very easy to bring all that data together and avoid, we didn't have any batch effects to correct. So the result is that it's a very high quality data set. And the reason why it's high quality is because we use the latest technologies such as 10x and NovaSeq. And we made sure to not pool the embryos. So we had to have this single embryo resolution, which is also important to obtain high quality. So we don't average out everything. One key aspect of our data set is that it's really the first single embryo, single cell atlas of development at early stages of development, before 24 HbF. So that's definitely something that I think is a contribution to the field that will help further analysis. And this is a very nice collaboration, again, as I mentioned. So one key aspect was to actually optimize the single embryo dissociation protocol, and it was really important because if you want to use the 10x, you need really have to have a relatively high concentration of cells. Otherwise, it doesn't. You can't really proceed with experiments. So previous works would pool the embryos, and you would pull a lot of embryos together, and then proceed with the cell dissociation protocol. And so you would end up with data set that were the result of many embryos pooled together. But even that would not really result, in our hands at least, with high enough concentrations of cells for our purpose. So that's even less so the case if you have just one single fish or embryo. And if we basically with our new protocol, which you'll see in our preprint, with our improved protocol, we could actually get from a single fish much more cells per microliter, and basically make it possible for proceeding with our experiments. And here's the website that we designed with the data science team here at the Biohub, that lets users, anybody really in the world, to explore this data. You can highlight different clusters and see what are the names of these clusters. You can zoom into a part of the UMAP and try to understand the expression profile of different genes. And here, for example, I'm interested in a region of cells that are surrounding the tail bud. So I just surrounded that. I can zoom into that, and then try to see what can be understood here. And so here, for example, one thing I do is I create a gene set, and I will mark two. I will choose two genes, SOX2 and TBXTA, that are related to markers for mesoderm and ectoderm. And then I can see here that there's an interesting population of cells that co-expresses both, and perhaps these are NMPs. We'll discuss that in a second. But yeah, very interesting. You can do a lot of explorations. That's just an example, but you can basically explore the whole data over all time points at a time, or one development stage at a time. So it's very full of insights, and we hope that people really will benefit from this. as much as we've been benefiting from it. So where are the coming back to an NMP question, where are the neuromesodermal progenitors in this picture? Well, as I mentioned already, there's two markers, SOX2 and TBXTA, that are good markers for the mesoderm and the ectoderm. So we can use them as proxies to try to identify the NMPs in our maps. If we if we look at the region in the UMAP that co-expresses both, and if we zoom into that, we see a population of cells here shown in red, in yellow, that seems to bridge a number of different tissues. You have at the top of the picture here neural tissues, neural posterior hindbrain. You have the floor plate, notochord towards the top left, and then below on the other side you have the presomatic mesoderm, PSM, lateral plate, and other mesodermal tissues that are more differentiated as you move along that trajectory. So very interesting to see that really it seems that the region that seems to bridge these different tissues potentially is actually where both of these markers are co-expressed, and it's really the case. So if we zoom into it, we not only see that this is a region that co-expresses both again, but that there's a gradient between TBXTA and SOX2, suggesting that the cells that are closer to the neural tissue actually express more SOX2 than TBXTA, and conversely, the cells that are closer to the presomatic mesoderm express more TBXTA than SOX2. So really suggesting there's a gradient there. But we can go further. Actually, we can use RNA velocity to try to understand how the cells are flowing in that space. So there's this technique, RNA velocity, that's very established now in single cell sequencing. It's really in the toolbox now, and we can use that to understand and to understand how are cells flowing in these maps. And this comes from splicing. We can basically, by comparing the spliced and unspliced RNAs, we can use that as a way to get a sense of the rate of production of the different genes, and therefore compute a vector field that will basically suggest in which directions are the cells flowing in that state space, in that transcriptomic space. So we can do that for the whole UMAP. We can do it here on the UMAPs surrounding the NMPs, and we can zoom further. And what's really remarkable is that we see that the NMPs seem to be at the source of many cells. So it seems that the NMPs from this population of cells that we presume are NMPs, everything flows from them, and the tissue and these cells go towards more precisely differentiate into mesodermal tissue at the bottom, at the top right towards neural tissue, and perhaps even floor plate and notochord. So that's really, really interesting. And we could not have chosen a more interesting population of cells. We actually never saw this before choosing to work on NMPs, but it turns out the NMPs are really turn out to be a very important population of cells in this picture. And so perhaps perhaps it was not a surprise, because the whole community found these cells extremely important, and I think this confirms that they are definitely doing something very special, because that picture where these cells are kind of the source of many other types of cells is not something you see everywhere in the UMAP. So it's like clearly a very important population. And so we can ask, because our data set, our single-embryo single-cell sequencing data set is temporarily resolved, we can also ask the question of what are the fate dynamics of NMPs over time. So how does their fate changes as time progresses during development? So if we essentially look at each data set for each developmental stage in isolation, we can look at this flow of cells, at these zoomed in UMAPs and the RNA velocity vector field, and we can try to understand what's happening and how that's changing over time. And so here, very clearly, again, at early stages, at bud stage, the NMPs give rise to both neural and measurable, and you can really see the population between these two fates, and clearly it seems that NMPs go to both neural and measurable tissues. They give rise to both neural and measurable tissue, but as time progresses, and we see a transition where suddenly the NMPs now seem to give rise to mostly cells that then integrate into the mesoderm, or at least that's the suggestion from the RNA velocity. And of course, there are lots of caveats because this is not, this is not a direct observation of a fate, but clearly it seems from our data that as time progresses, NMPs remain attached to the mesodermal manifold and detached from the neural one, right? And this becomes very clear as we progress during development. So that's very interesting. So that would suggest that early on, they are actually behaving as bifated progenitors, but later on, they lose that dual fate and now are exclusively giving rise to mesodermal tissue. So we can quantify that, and it does appear that that's the case. You can look at your maps and interpret them, but there's lots of caveats when you interpret a projection from a high dimensional space. But we can also do measurements that in this original space and try to understand the transition there. And clearly, our data suggests that as we progress during development, as we go from bud stage to 30 cell mites, there's clearly a shift towards a measurable fate. And essentially, we can look at this RNA velocity field and see that basically the NMPs are mostly going towards the mesoderm at later stages. So the current picture now is that NMPs are bifated progenitors only early in development. Later on, they lose that dual fate. So that's our hypothesis from the single cell data. And the question here is, can we validate these findings with imaging? Because remember, my team is also building these advanced microscopes and developing tools for processing images and gather insights from them. And so can we now go back to imaging, and then try to see if we can observe this transition in the fate of the cells from a bifated to single-fated. So we had, of course, as I started my lab, we had to build some very, very, very advanced microscopes. I was lucky to have worked in a postdoc with Philippe Keller and his team, and also with Jean Myers in Dresden. And I was very lucky to learn how to build these very complex systems. And so we built a very nice, multi-view light sheet microscopes that basically has 4 objectives surrounding the sample, capturing a total of 4 images, and we can do that within 30 seconds. And the sample is mounted in a cylinder of agarose, and so that we can actually really look at it from all these sides. And this is really important because it's difficult for light to get in and out. And so if you have multiple points of view into the sample, you can actually generate these very beautiful movies that you've seen already. And so we also added some new capabilities to this microscope. We added a photo manipulation arm that allows us to photo convert Dendra2. And so by doing this, we can mark cells of interest at different stages during development and then follow them. And it's much easier to follow cells that you've marked than to try to follow all the cells. And because of the sparsity of that labeling and because of its separation from all the other cells, it's much easier to do so. And also to speed up imaging, we have a strategy where basically we follow the cell. We have a temporal sampling that's finer for the photoconverted cells than it is for all the other cells. So we keep the context by imaging from time to time all the cells in the embryo, but we focus our temporal resolution on the cells that have been photoconverted. So this is very nice work that that Merlin in my team has done. He's actually the first author on this on this NMP project, and it's very so this really so we did this experiment at birth stage. So we photoconvert the NMPs where we think they are, and we think we know where they are because we did some HCR experiments where we basically figured out at different stages in development where are the cells that are co-expressing SOX2 and TBXTA. So from that we could map out their localization, and then this is of course all fixed because of HCR, but then we can use that to basically know where to photoconvert the cells precisely. And so once we have that knowledge, we photoconvert exactly where we think the NMPs are, and then we can follow them in time and space. And so here's the video where you basically proceed during development, and then you can already see that there's quite a lot of cells that are still magenta. So magenta is photoconverted, and cyan is all the nuclei. So we look into more details here. We can clearly see there are photoconverted cells here in the red, both in the neural tissue and in the somites. And so that's really suggesting that it seems to confirm the idea that the hypothesis we got from single cell sequencing, that early in development, at birth stage and a bit later than that, the NMPs still have this dual fate. But then we do the same experiment later at 10 somites, and then we want to see if that is still the case, and if they still retain that dual fate or perhaps not, as we've already hypothesized. So if we do the imaging, and we can see if actually these cells will actually give rise to both tissues or not. So we run the imaging, and then at the end we can already see here that most of the cells are in the somites. So on the top side and the bottom side, right here and here. And so if we look at it a little bit more in detail, the neural tube has no cells, right? And I will show you more data that is perhaps a little bit more convincing later. So it's clearly, so we have done a lot of experiments and we have done quantifications, and the pattern is clear. At later stages, the cells that we photoconvert do not give rise to both tissues, and we most likely are actually photoconverting more cells than we should, and so the NMPs are probably a subpopulation of the cells that we're photoconverting here. But if anything, that would give rise to potentially more cells that would be photoconverted than we want, and still we can't see cells that give rise to the neural tube when we photoconvert at later stages at 10 somites. So clearly, we think we have confirmed our hypothesis. The next step is, can we actually see these individual NMPs? Can we actually follow these cells? And so we've been trying, and it's very hard. It's very hard. But we have here a very nice example of a cell that we can track. It divides once here. We keep following the two descendants. And again, this is, we're still pushing the limits of imaging here. And, it's extremely hard to follow this. But this has been checked by hand. So we have, relatively high confidence that this is actually real. And so when you look at it at the end, we clearly see that the two cells give, these two daughter cells have actually integrated, one in the differentiated mesoderm in the somites, and then the other one in the neural tube. So that's very exciting, because that's perhaps the first live visualization of an NMP, and then we're working on having more such data. But what we also realized is that we needed an even better microscope. And so what we did is we went back to the lab and started building something new. And so that's where Bin Yang in my team at the time, he since last November, he left, but he while he was in my team, he did a fantastic work to build for us a fantastic new microscope architecture, which we call Dashi. And this is in Mandarin Chinese, Da means large and Shi means fine. And the idea is it's a microscope that can image at very high resolution, both spatially and temporally, but also over a very wide field of view and over a very large imaging volume. And it was published earlier this year in Nature Methods. So here's a beautiful animation that was produced by a very famous Twitter addict, Lashi. He's known for his very nice videos on microscopy and nightmares. And so he has designed this very nice video that explains that if you have a microscope with lots of objectives surrounding your sample, it's very difficult it's extremely hard to image your sample in a practical way. So you want instead to have a single objective in contact with the sample or in contact with the thing that holds the sample. And then if you do that, then you have to have an oblique light sheet. And if you do that, then you can actually potentially do high throughput imaging because it's much easier. It becomes more of a traditional microscope, an inverted microscope. And it's much easier to handle the imaging, right? And so, of course, it complicates the design to have an oblique light sheet. You need a lot of optics to be able to do that. But, and it's particularly, you need a special objective that allows you to do that, right? But I won't go into too much details here because the audience is maybe more biologists than physicists, but with the resolution we can achieve is really high. And I think we can, for these kind of imaging volumes, we can probably push it even further. It's about 400 nanometers laterally and 2 microns in depth. And so, as I said, we collaborated with this team at Calico from Andy York and Alfred Milik-Seeking in his team to build. They designed this very nice Naughty objective that we're using. We had one of the first versions of it, and that made it possible to actually to build this microscope with the kind of optical capabilities that it has. And because of this inverted configuration, and because if there's only one objective in contact to the sample, and because of the obliqueness of the light sheet, we can do this high throughput imaging and have multiple embryos imaged sequentially, which is very hard to do. Not impossible, but very hard to do with a multi-view light chain microscope where you have at least typically 2 or 4 or more objectives surrounding your sample. And the kind of data we can get from that is really exquisite. You can cover very large volumes at very high temporal resolution here, 30 seconds per stack. We have 2 views. I can't go into all the details of this microscope, but we have we can actually send the light sheet over 2 different angles and collect at least 2 views in the sample. And you can see here in this zoomed in views that you can actually follow almost all the cells and especially see the divisions of the nuclei. Well, what you're seeing here, really every dot is really the nuclei of every cell, right? So when I say a cell, it's really what you're actually seeing is the nuclei of the cell. And with some collaborators here at UCSF, Tom Kornberg and his postdoc Van Peng Wang, we looked at other kinds of samples with the same microscope, and we showed that you can image, because of some of the features of our microscope, we can image up to 10 millimeters of tissue along one dimension and still retain very high speed and high resolution. And so this video here is I think more than 4K. And it's kind of, it's even difficult to look at all the pixels on the screen, even if you have a very high resolution TV, which is just phenomenal. And of course, this generates terabytes of data very quickly, right? These machines, especially our latest ones, is definitely producing a lot of data. But generating images is fantastic, but you want to actually generate insights from them, right? So you need to process them. And so Jordan, Jordan, my team has been working on a tracking algorithm, and we've been taking some of the off-the-shelf ideas and being pushing them to the limit and adapting them to our needs. And so here you see a time lapse from our latest Dashi microscope, right? And now you see the segmentation of all these nuclei. And then next you will see essentially the tracking of all these nuclei. So, so that winds back the time, and then we'll play it again. And so here you see all these little tracks are essentially the trajectories of these nuclei that essentially are proxies for following the dynamics of all the cells in the developing embryo. And if you really look at the tail bud, so on the left side it's the head, but on the right side bottom it's the tail bud. You'll see it's very dynamic. The tissue is moving very, it's very messy, and that's because the cells are moving very fast. And so, yeah, so we have access to all this data we can track. And actually, this is already a 1-year-old data set. So, we have actually better tracking by now, much better tracking, actually. And so what we can use with that, we can actually do some sort of in silico experiments. So we can use all this tracking data and follow the cells. And one thing we can do is we can do the same experiments we did in the microscope by photoconversion, but in a completely digital fashion, and ask the same question. Are these presumptive NMPs integrating both mesoderm and neural tissue or not? And at which stages do we have dual fate, and at which stages do we have a single fate? And so if we actually do this, we actually can see that early on you see cells that integrate both the neural tube and the mesoderm, but not when you do it later. So when you do it later, the cells do not cross this orange boundary that separates the somites from the neural tube here in this projected view. So that's another, we have quantifications for it. That's another confirmation of our hypothesis that there's this transition in the fate of NMPs. We have much more, details in our upcoming preprint, but I think this is enough for now to illustrate, what we've been trying to do. Yeah. So that concludes my talk. I would like to thank you for your attention. The driving force for that project has been Merlin Lang in my team, who's a senior scientist in my team, who's been with me from almost the beginning of my adventure here at the Biohub. He has helped me set up Zebrafish as a model organism at the Biohub. Very close second on that is Shruti, who has been doing a lot of the single cell sequencing and dissociation protocol optimization. Hiro has been always helped, was a postdoc in my team, working on some other projects. Mason Campbell from the theory team at the Biohub has been helping with some early tracking. Amit Chansolak is a software engineer that has helped us on many projects. Luke Hyman was an intern for 1 year, was working on different projects. Michael Borja in the genomics team has really helped us a lot on the single cell sequencing protocols and pipelines. Norman Neff is leading the genomics team and has been a fantastic partner in that, together with Angela Pisco, who in data science, who leads a team in data science that has been helping us on all the single cell sequencing analysis. In particular, Alejandro in her team, then Alexander also, and Aaron at the Biohub have been also helping a lot on the image and on the single cell sequencing data analysis. Bin Yang in my team has helped, has built this Dashi microscope and also the earlier system. Sheng Zhao is a biologist in my team working on different projects and has helped also on Zebrahub in terms of annotation. And Jodao has done all this tracking you've seen here. Rafael Gomez-Roberg is director of bioengineering, has been helping us a lot on some of the designs of our microscopes. Yeah, that concludes it. And one last thing I would like to say is Olivier Pourquier and Sharon Guillot have been really guiding us a little bit on kind of pointing us to this question with MPs and telling us, look, try to look at these cells and see what you can find. And I will thank them for this very, very good tip, because the NMPs turn out to be a very interesting and fascinating population of cells in early development. Thank you again. I'm still online.
All right. Thank you so much for your talk. Those are mesmerizing images for sure. Those are beautiful. So we have a few questions here. Actually, we have one question from the chat. It's a question from Nicolas Trigueiro. He's asking, is the data available on the Zebrahub site already pre-processed for mitochondrial genes expression and counts, etc.? He's saying that he's currently trying to use this data in his PhD.
Well, I think we have all the data. You can download all the data on the website. So it's not just it's not just interactive visualization using cell by gene, which will developed by CZI, but also we also offer the direct download of the data. For mitochondrial genes, I'm not sure we have done anything specific on that, but there's a way you can reach out. You can contact us on the website, and so if you have questions, the best way is to contact through the form that's there, because then also the data science team will see your question and maybe the experts can address the question for you. But yeah, we're definitely interested in helping people to leverage our data.
Perfect. So another question from Fabio Zolessi. He's saying it's wonderful work, so important for cell development biologists. Yes, totally agree. What resolution can you actually get with this microscope? Subcellular structure like cell processes? What's the acquisition lens NA?
You're asking about the resolution of the microscopes?
Yes.
I mentioned that it's about, let's say, to be conservative, it's about 0.5 micron laterally, and then 2 microns axially. That's for the latest microscope we have, and we can do that over 10 millimeters maximum along one axis, 200 or 300 in depth, and about 800 microns laterally. So that's a fairly large volume. So in terms of voxels, we usually have is easily 6,000 by 2,000 by 400 also pixels.
Wonderful. So I have one last question, if you allow me. So
yes.
These NMPs, they're very interesting subjects. I was just curious if have you ever tried to investigate them on mice, for example, or other animals during development? Do they behave in the same way?
Oh no, we only have done NMPs in Zebrafish, only Zebrafish.
Okay, perfect.
Yeah.
So Dr. Loïc Royer, thank you so much again for being here with us and sharing your amazing data. Congratulations. I really think they are mesmerizing. I really think they are beautiful. Thank you so much, and congratulations to you and all your group.
Thank you for your attention, and thanks for inviting me today to present this work. And you should be seeing the preprint, very soon on bioRxiv. Thank you.
Perfect. Thank you, and everyone else. So we're gonna have a break right now, and we will be back with the Global Education Network on Zebrafish at 3:30 p.m. All right. Thanks a lot, everyone.
Bye-bye.
Bye-bye.